So what I'm gonna talk about today is drug repurposing strategies for COVID-19, a bit of an overview, but also some of the work we have been doing at Essex. So there you go. So a couple of things about me. So I did my undergrad and master at University of Siena in Italy. And funny story, I graduated during the SARS 2002-2003 pandemic. And actually my work during my research project was on SARS. I then moved to do my PhD at the National Institute for Medical Research in London. And after that, I did a postdoc in Imperial College, and I started working on cancer research. And then in 2015, I opened my independent research group at University of Essex, where I still work on structural biology, mostly applied to cancer research. But I'm back to biology as well now. Couple of things about our department. So I work in the School of Life Sciences, very nice and interdisciplinary research environment. We have quite modern facility. The one you see here is actually, I'm gonna put my laser, actually. There you go. The one you see here is actually our brand new STEM center. We were extremely proud to have attracted 4.5 million in research income last year. We have about 1,000 students and 6 bachelor courses and 6 master courses. So why do we do a career in science? Why should we do a career in science? I think, especially now with this pandemic, it's quite clear why it's so important to have science research. But independently from that, these are just some data from the last 4 years about the increase in demand in the scientific sector. So as you can see in the industry, there's a 20% increase in demand in jobs, 4% in hospital. And as you can imagine, these numbers are probably gonna go up massively in the next few months because the sector, pharmaceutical sector, is actually highly active right now. For the teaching side and the research project with my students, I'm mostly involved with several of the masters that we offer. At Essex, we have the Master in Cancer Biology, in Molecular Medicine, in Biotechnology, in Data Analytics, specifically applied to Health Genomics, Tropical Marine Biology, and we have a brand new Master in Structural Biology and Drug Design. That, as you can imagine, this is quite relevant for my talk, but this is going to open next academic year, not 2021, 2022. One of the things I am most involved with master students is the independent research project. So all our research students have to do an independent 90 credits module in which we're going to have to do a real research project for 20 weeks in a lab. And they have to do all the science and then the writing of a report. But not only, they have also to present the data during a research conference. The type of subjects that the students can work on are protein biochemistry, enzymology, structural biology. This is the type of work they mostly do in my lab. And these type of techniques can be applied to study, for example, virology, cancer biology, or stem cell. There are also, we have also a quite strong group in data science, genetics and genomics, and social genomics. This is quite a cool hot topic right now. But not only, we work a lot in genetic engineering, biotechnology, microbiology, molecular biology. So there's quite a lot of different subjects that the student can choose to do their independent research project. But not only that, the students can do their research project also in industrial setting. So they can go and work for 3 months in the labs of one of our industrial partners. Here are some of our main industrial partners. Very quickly, I want to point out the very key feature of the things we do during our master. All our master have a module in gene technology, synthetic biology, genomics, and protein technology, because we believe that these are the key components that every master student should have, every master student should know. Then, according to the master, there is a core module specifically for cancer biology, we have the advanced cancer biology. In this module, we cover topics like molecular oncology, cancer epigenetics, which is extremely relevant right now, is a very big field of research right now, tumor microenvironment, but also novel treatment for cancer, like stem cell treatment or immunotherapies. We have also clinicians coming to the university to give lecture. And this is quite important, so our students are absolutely updated to the type of treatment and diagnostics that are available in cancer research and in the hospital. The molecular medicine master, the core module is molecular mechanism of diseases. And this is quite a broad type of module. We cover microbiology, immunology, and virology, as we can imagine. But not only that, we also cover topics like prion amyloids, which are quite relevant for the understanding of how neurodegenerative disease are originated. Similarly to cancer biology, we also have expert and clinicians coming to the university to give lecture, so the students are updated directly from the people on the front line. The master in structural biology and drug design, the core module is actually going to be called structural biology and rational drug design, and we are mostly going to focus on the use of structural biology to drive the design on novel drugs. We're gonna cover experimental structural biology, but also bioinformatics, and this is quite relevant for the things I'm gonna talk about during my webinar. We're not just gonna cover that; we're also gonna cover novel methods for development of drugs. For example, chemoinformatics, computer drug design, but also fragment-based drug design. I don't really want to spend more time as talking about our courses, but if you want more information, here you got this link, and you can chat directly with staff and students, or you can chat with me and other members of staff and ask directly any question that you have. But you can also just contact Uli or me directly using our emails. So that was it. And now I want to enter into the topic of this webinar. So in the first part, I want to talk about the SARS-CoV-2 infection. I'll give a bit of an overview. I'm going to probably spend most of the time talking about that. It's quite complicated. But this is quite helpful because give an understanding of what are the things that we can target with drugs to prevent infection. So I'm going to give a bit of a quick overview about drug targets. This, as you can imagine, is an extremely vast subject. There are tons of paper coming out every day talking about drugs or new clinical trial. Just to give you a quick spoiler, there are over 300 clinical trials for drug testing and vaccine testing for SARS-CoV-2 and COVID. So the subject is vast and it's quite challenging to keep track of all the literature that is coming out. But I'm going to try to summarize a few key points, and then I would like to show you some of the work we have done on COVID at Essex. So what is coronavirus? This guy here, it's a coronavirus. So the reason why we call them coronavirus is because this type of virus at the microscope looks like a crown. They have a crown shape. So you see these spikes that come out of the surface of the virus, they have a crown shape. From this crown shape, the name coronavirus. They are RNA viruses, so they have a small genome, which is made of RNA. And this give a lot of opportunity to the virus. It's a very flexible type of genome, is subject to constant changes. And this type of changes, mutation, have the virus to adapt, and it's what allows the virus to infect new hosts. We have to say that for SARS-CoV-2, there's not been, is not mutating too quickly, so there hasn't been reported many dramatic mutations, but let's say that the virus, it's however equipped to do that. So it's equipped to change rapidly. There are currently 7 known coronavirus that can infect humans. 4 cause common cold. So the colds that we got every season during the winter, 50, they are all viral colds, and 50% of them are caused by coronaviruses. So they are, our bodies adapt to respond to them. However, there are three which our body is not quite equipped to respond in an efficient way. And these are the SARS-CoV-2, which caused the pandemic nearly 20 years ago, 2002, 2003. MERS, which is another pandemic, for coronavirus related, and the current one, so SARS-CoV-2, which is the virus, which caused COVID-19, which is the disease. The coronavirus, the virus, enter our body through our nose and mouth. So we breathe the virus in, but that's not the only way in for the virus. We can actually, the virus is quite sticky, so it can stick to our hands, and then we touch ourselves in the mouth, nose, and eyes, and through this entrance point, the virus can enter and infect our body. The good news is that in 80% of cases, the disease is quite mild and does not require hospitalization. The symptoms are quite nasty, but the disease is not. We call it mild disease. In 20% of cases, the people are affected by more severe outcome, and they develop pneumonia, which is the most common one. The harmful part of this outcome is that this disease prevent the oxygen from going from the blood to the bloodstream. So what happens is that the amount of oxygen diluted in our blood is drastically reduced, and that's quite bad. However, the most common reason of death connected to COVID-19 is actually not directly connected to the virus, but is due to our immune system. So it's actually an overstimulation of our immune system that is causing damage to our organs. So this overproduction of this early response of pro-inflammatory cytokines, which is sometimes referred as cytokine storm, is actually what is triggering this massive inflammation, which is causing organ damage and organ failure. So the things I'm going to talk you quite in details about is what happens to the virus. So the virus arrives, enters our cells, replicates, and then from one cell, lots of other viruses come out. So these I'm going to go into quite detail here. This is the way our body responds. So the virus comes out, it gets ingested by the antigen-presenting cell. The antigen-presenting cell will present on its surfaces this antigen, which are recognized by the T cell, and the T cell will stimulate the B cell for producing antibodies, will coat the virus and prevent the virus from entering our body, our cell, and then will stimulate other immune cell, which will kill the infected cell. So this is the way our body normally replies to the infection and get rid of the virus from our body. I will not talk about the vaccine because we can do a 3-hour webinar on vaccination only for coronavirus. So I will try to talk about everything but vaccination. So this is the way the virus actually looks like. This is a schematic representation of the virus. The virus is enveloped inside a membrane, which is this light blue ring here. So it's a kind of a sphere, and inside of this sphere, you've got the genetic material, which is RNA. This ball of membrane is actually a ball of fat, and that's the reason why soap killed the virus, because the soap dissolved the fat, open this envelope, and the virus died. So that is why we need to wash our hands quite frequently, and that's why soap is actually quite effective in getting rid of the virus from our hands. On this envelope, we got 3 different proteins. One is the spike protein, one is the matrix protein, one is the E protein, envelope protein, and inside our of the virus, we got the genetic material, so the RNA, and the RNA is wrapped around this protein, which is called nucleocapsid protein, which keep all the genetic material nicely packed. The matrix protein has been shown to be quite important for the shape of the virus. So it's important when the virus comes out of a cell to have a nice shape and nice structure. And the M protein is kind of like the organizer of the shape of the virus, because binds all the other protein on the envelope. The M protein, as I said before, is bound to the genetic material, and what is important, it keeps all the material nicely packed. And what is also important is important when the virus enters the cell, helps the RNA to get transcribed and assembled. The E protein is very small, it's here, and what it does is been shown to be important for the production and maturation. So it's important when the virus comes out of the cell. The spike protein is indispensable for the virus to actually enter our cell. So as I said, all the virus is coated with this spike protein. What the spike protein does, it recognizes the receptor on the surface of our cell, and it binds it. This receptor on our surface is called ACE2. There's a lot of ACE2 on the surface of our cell, and especially on the surface of our cell in the lungs, which clearly is the reason why the virus enters through our lung and breathing system. Once the virus is bound to the receptor, then another protein arrives. One thing I wanted to say, ACE2 has a function in our body, is important to lower the blood pressure. But what is interesting is that this actual function of ACE2 is absolutely not related or required for infection. And I'm going to show you that this is quite relevant when we think about drug targeting. Once this ACE2 is bound, arrives another protein that is on the surface of our cell, which is called TMPRSS2, this guy here, quite a mouthful of a name. It's quite an interesting type of protein because it is very commonly associated with ACE2. They localize on the surface of the cell together, so there seems to be body, they're always together. What this guy does is chopped the spike protein in two pieces. This chopping allows the spike protein to mediate the fusion of the membrane of the virus with the membrane of the cell, and this allows to inject the genetic material of the virus inside our cell. What is interesting, it's been shown that this method of entry of the virus in our cell is exactly the same of the SARS coronavirus that caused the pandemic in 2002-2003. Now, the replication system inside the cell is quite tricky and quite fascinating on the complexity and organization that in which it works. So the first steps is the viral RNA enters inside our cytoplasm of our cell and is floating around. It's floating around, and then eventually it binds our ribosomes. And our ribosome binds this mRNA and they start producing protein. Specifically, what they do, they produce two big proteins, which are called polyproteins. The polyproteins per se don't do anything. What happens is that two viral proteases called PL-Pro and 3CL-Pro arrives and start chopping this polyprotein and create lots of different proteins. So basically what they do, they activate all the viral proteins of the virus. The second step is at this point, all the viral proteins have been activated and they start working. This protein, for example, one of these proteins is one of the most important, is the RNA-dependent RNA polymerase, which is this guy here. So what this guy does, it takes the genetic material of the virus and reads it and creates a copy. So the genetic material of the virus is a plus sense RNA. What this protein does, it creates a minus sense RNA. So why does it do that? It does that because it's quite useful, because from this minus sense, this protein is able to produce, this minus sense is used to produce 4 protein, the N, M, E, and S protein we saw before, but not only, the polymerase itself reads the minus sense and create a plus sense. So at this point, we go the 4 structural protein and the genetic material, which has been copied and duplicated. Now this, the membrane envelope and spike protein enters our organelles in our cell, specifically the endoplasmic reticulum and the Golgi system. So what we have the nucleocapsid protein that binds the newly formed genetic material of the virus and create this nicely packed genetic material of the virus. Then this genetic material of the virus recognizes these organelles, which have all the viral protein sticking out, and it binds to these protein sticky notes. And what happens is that these vesicles, these organelles, our organelles, which has been coated with viral protein, go envelop this viral RNA and create this form of vesicle. So it's a membrane, and inside this membrane, there's a newly formed virus. This vesicle reach on the membrane of the cell, fuse with it, and release new virus. So starting from one virus, the virus enter, duplicates, create new protein, new genetic material. The new protein allows the virus to get assembled, and our cell start freeing lots of new viral particles. So, so this is the way it replicates. Why did I go from this very complex system? Because there are, I wanted to point out lots of different proteins, which are key steps for the viral replication, which we can target for drug studies. So the first thing is, why don't we use the antiviral drugs we already have? The reason is that the antiviral drugs, or the most common antiviral drugs we have, it's like Tamiflu or Lenza, they are neuraminidase inhibitors. So neuraminidase is this guy here, and they are transmembrane protein present on the viral particle of the flu virus. So if you see the schematic representation, it looks quite different from the schematic representation I showed you before. And the reason is that the protein which are on the surface of the virus of the flu are quite different from the protein which are on the surface of the virus of the coronavirus. Specifically, the flu has two classic proteins, hemagglutinin and neuraminidase, and the coronavirus doesn't have these two proteins, which means that these two drugs, which target this guy here, will do absolutely nothing to the coronavirus. So what are the therapeutic options? So as I said, the protein which are really key for the virus replication are the spike protein, which is indispensable for the virus to attach on the surface of the human cell and enter inside the cell. The ACE2 and the TMPRSS2, which are on the human cell, which are mediating the attachment of the virus, and they literally bring the virus inside the cell. The proteases, which is required to activate all the viral protein, and the RNA polymerase, which is replicating the virus itself. So if I shut down any of these proteins, I block viral infection. So what is the idea? The idea is that we are in a time of pandemic, so we don't really have the luxury to start a drug development project from scratch, which takes years. So we are in a situation of urgency. So the idea is, can we use something which is already on the market, but can be useful to treat coronavirus? This concept is called drug repurposing. What does it mean? Basically, it's a strategy in which we identify new uses, new medical benefits of drugs which have been already approved and are already on the market. But these drugs were originally developed for something different. Let's make a classic example, Viagra. Viagra was actually developed for a completely different purpose, was developed for a blood pressure related use. But in reality, one of the side effects during clinical trial was reported to be better of the effects of the first medical indication. And that's one classic example of drug repurposing. So they simply rebranded the drugs, and that is a strategy for drug repurposing. However, in the vast majority of cases, drug repurposing happens like what happens for Viagra. So an accident during the development of the drug. Suddenly, we don't really have a luxury to wait for an accident to happen. So what we need to is actually to investigate in a much more systematic way and identify if there is the option of finding new use for already approved drugs. There are lots of advantages for this. As you can imagine, there's low risk because most of these drugs have already approved. So from a safety and toxicology point of view, they're already all have done through approval. So we know that that is not an issue. There's a lower cost of development from a time point of view because these drugs can already go in clinical trials or already in preclinical trials. And it's much cheaper because there's process of developing drugs is much is much reduced. Just to give you an idea, and as you can imagine, in time of pandemic, money is an issue. The cost for repurposing a drug is about $3 million, while a classic drug development project costs about $3 billion per drug, so massive reduction in cost. So what is the idea? The idea is that we take all the drugs that have been approved, we put this drug in a software, and we allow this software to fit the drugs on the structure of the protein. If we find that some of these drugs fit nicely and prevent the viral infection, then we need to test this drug further. So we take all the drugs, we use the software, we use the structure, we check if these work, and then from the scoring results, we see if this is an option that will work or not. However, there's a limitation, because once we have identified a good target, what do we do with that? Now, how do we know that this drug is actually working in viral infection? Are we going to test directly in humans? Well, that is a bit tricky, because we can't really infect healthy people to check if a drug is working or not. And also, while this drug we know is safe and doesn't have toxicology issue, it's also true that these drugs will require to be optimized, so we need to optimize the dosage. So how do we know that the dosage required for blocking infection is not actually toxic? So we can't really jump directly in clinical trial. So we need an animal model to study all these drugs that we identify with this bioinformatics approach. But which one do we use? You could be surprised, but this is an issue that has always been there since the first pandemic of SARS in 2002, 2003. The animal models have always been an issue. However, due to the fact that the SARS in 2002 didn't infect that many people, only 1,000, there's not been much research on it. So the issue is that despite that, we still don't have a very clear understanding of which is the most suitable animal model. So what we did in our lab, in my lab, in collaboration with Greg Brook at Essex, was to simulate using computer software the binding of the virus protein to the receptor. And we simulated this binding for the receptor of human by also mouse, cat, dog, rat, guinea pig, ferret, and rabbit. So what we saw is that the SARS-CoV-2 spike protein has really evolved and optimized to bind the human protein, and it does it much better than SARS-CoV-2. And this much better binding is actually related to the fact that the SARS-CoV-2 has a much higher infection rate compared to the SARS-CoV. But not only, we have realized that this binding is optimized for the human one, but is really not optimized for rodents. So mouse, rats, and what was that? Guinea pig really don't work that well. The binding is not really that efficient. It really doesn't work that well. So they are really not suitable animal model. So the only animal which has the most similar binding mode than the human one is actually ferret. So according to our study, ferret seems to be the most suitable type of animal for animal studies of drugs. So what I would like to do now is to get a very big short summary of what are the main drugs that have been tested at the moment in clinical trial. So as I said, the protease is indispensable because it's required to activate all viral protein. What does that mean? That means that if I shut it down, the virus will enter our body, it will enter our cell, but will not be able to replicate. What is interesting is that this protease is completely different from any other protease that we have in our body, which means low toxicity. So drugs that block this protein very likely will bind nothing else in our body, which is good. The two ones that everybody was talking about 2 months ago were the anti-HIV, antiretroviral drugs, lopinavir and ritonavir. So lopinavir is a protease inhibitor, is a viral protease inhibitor, while ritonavir increases the half-life of lopinavir in our body. Sadly, some clinical trials on these drugs are already coming out, and the results of some of two small clinical trials show that these two drugs actually don't really have much of an effect on the duration of the disease. This is not too surprised, because if we actually check the structure of the HIV proteases and the SARS proteases, already without going too much into the details of the actual structure, they look very different. So this is actually the virus, HIV viral proteases with the lopinavir inhibitor bound to it. And as you can already see, the way this inhibitor binds, the position in which binds, and the shape of this binding is completely different from the site in which the inhibitor binds the SARS virus. As you can see, this is the site in which the protease needs to bind an inhibitor. And this is another inhibitor called N1, which seems to be a bit more promising than the HIV virus. So again, this was not a good result, but not too much of a surprise. Drugs which is actually much more promising from that point of view is these alpha ketamide inhibitors. What they were originally designed as broad-spectrum viral protease inhibitors, and they were this guy here. And what this group did was realize that these drugs can still bind the SARS coronavirus protease, but not that well. So what they did was to optimize this drug, and what they did using medicinal chemistry, so a bit of modification of this compound, they produce this guy here. And what they showed is that this guy here is actually very efficient to shut down SARS-CoV-2 proteases. This was actually broad-spectrum proteases, so it was able to block quite a lot of proteases, while this one seems to be more specific for SARS-CoV-2. It doesn't block anything else. So they sacrifice the broad spectrum for specificity. And what they show is that mice are, from a toxicology and survival of the drug inside the bloodstream, this drug is quite well tolerated by mice. And this should enter clinical trial quite soon. The other protein that is very interesting from a replication, from a survival of the virus point of view, is the RNase-dependent RNase polymerase. So now, as I showed you, this protein is indispensable for the replication of the virus. So if this protein is not there, the virus does not replicate its own genome, which means that new viruses are not produced by the cell, which is great. What we notice by looking at the structure of this protein, this is the SARS-CoV-2, this is the Zika virus and hepatitis C virus. Zika and hepatitis C, like SARS, are RNA viruses, and they all have an RNA polymerase like the SARS-CoV-2. If we check at the structure, we notice that the overall architecture is quite similar. The overall architecture is similar, they bind similar things, they have to produce RNA starting from an RNA template. So their way of working is similar, the structure is similar. What does this telling us? There is good chance that the inhibitor already designed for hepatitis C may work even for SARS-CoV-2. And what has been shown is that inhibitors for these hepatitis C polymerase actually work decently well even on the Zika virus. So what has been done was to, this was actually a very early stage type of clinical trials, ribavirin and remdesivir were put in clinical trial pretty much immediately as soon as the pandemic started. And now there are two more RNA polymerase inhibitors, which are called favipiravir and galidesivir. So the clinical trials results for ribavirin, remdesivir, some are already coming out. Now they are a bit contradictory, and the benefits is not quite clear. The reason why ribavirin is not quite clear is that it was used as a cocktail of drugs. So it was a combination of 4 different drugs. And it's not quite clear, the cocktail seems to be promising, but it's not quite clear if ribavirin is actually doing anything good in that cocktail. So it's not quite clear if its contribution is important or not. Remdesivir, again, is quite unclear benefit. There are quite contradictory results coming out of different clinical trials. it seems that it may have some improvement in the duration of the disease, but definitely has no improvement on death rate, which is probably the most important thing. And these other two drugs are currently in clinical trial, and the results are expected in July and August. So this was the main protein. The one I haven't talked about, which is one of the main protein, is the S protein, and that protein is the protein I'm working on. So the huge majority of vaccine are actually targeting the spike protein. And just to give you an idea, there are more than 90 different clinical trials targeting the spike protein with vaccination. Now, the idea is, okay, can we know that the vaccine will not be available for another year or so. So can we develop or repurpose a drug that binds on the spike protein and prevents the spike protein from binding the receptor? So basically, can we find a chemical compound that does exactly the same job as the vaccination? So that was our starting point. So what we did was to take 1,300 FDA-approved drugs. We used them, we used a docking software, and we docked this molecule on top of the receptor, exactly here. But we didn't limit to that. That is quite brutal. We just fit geometric fit. We wanted to simulate a bit better if these drugs had a potential biological activity or not. So we used quite advanced computational system called molecular dynamic simulation. And what we did was to simulate the binding and unbinding of this receptor to the spike protein alone and in presence of these compounds. And what we saw is that a group of compounds actually are quite efficient in preventing this recognition and this binding. And what we saw is that the majority of them belonged to a class of hepatitis C inhibitors. So it really is quite fascinating to see how these hepatitis C inhibitors have the potential to be effective even in the treatment of SARS coronavirus. And it's quite interesting to see how different inhibitors can block, different hepatitis C inhibitors can block different steps of the SARS coronavirus replication. So now we got quite a good candidate system, and the next step is going to be starting to test this inhibitor in vitro and then in animal models. I'm almost done. Just wanted to mention the last few proteins that are important for the viral replication. So these two are ACE2, the receptor, and TMPRSS2, which is the protease that activates the spike protein for the fusion of the membrane. So suddenly, ACE2 doesn't seem to be that promising for drug target. Why is that? So this is ACE2, this is the guy, this is our receptor in our cell, and this is the spike protein. Now, this is where the huge majority of drugs we got for ACE2 binds. They bind in this big cliff that is in the protein. This is a drug, for example. Now, the issue is that, as you can see already, this is quite far from the place where it binds. So it's quite challenging to design a compound that binds here and modify the structure of the protein so much to prevent this binding. So this is actually a bit of a dead end. Instead, TMPRSS2 seems to be way more promising, and what has been shown is that this camostat mesylate compound blocks the activity of this protein and prevents the cleavage of the SARS protein and the fusion of the membrane of the virus with the human protein. So these have been quite effective in vitro, and now there are, I can't remember the name, but the number, I think 5 clinical trials trying to study different inhibitors of this protein. So this is quite promising. The last one I wanted to talk about, because in the newspaper don't seems to mention anything but this hydroxychloroquine. Now, hydroxychloroquine doesn't target the virus. So hydroxychloroquine, what it does, it reduces the inflammation response of our body. So what this drug does, it doesn't prevent the virus from entering our cell. It doesn't prevent us from getting an infection. It doesn't prevent us developing symptoms. What it does, it reduces the massive inflammation response that our body does, and may prevent some of the most damaging effect of the inflammation in our body. However, I was reading in the last few days that these drugs have major side effect and neurotoxicity, and many clinical centers actually have stopped using this as a baseline therapy. So just to give you an idea of what is the difference between these drugs and the other drugs that we are currently trying to develop to target actually the virus. If you want to read more about this, I mean, there's a ginormous amount of literature coming out every day on this subject, so there's quite a lot, but these two Nature paper, I found that extremely informative and extremely clear and extremely accessible, and I really recommend everybody to have a look at that because they are quite informative and quite nice. Especially for vaccine, it really gives an idea of the amount of clinical trial for vaccine that is currently ongoing. Now, all the work that has been done on this has been done by my group Essex in collaboration mostly with Greg, still at Essex, and some of the work that we have done on the drug targeting of the spike protein has been done in collaboration with my collaborator at the University of Siena in Italy. And with that, I'm gonna stop here and I'm gonna open for questions.